You're risking losing a lot of marks on Biology Paper 1 if you don't know everything about this practical. I'm talking about the amylase practical. Here's everything you need to know. Firstly, the purpose of this practical is to investigate how changing pH affects the activity of the enzyme amylase. In case you've forgotten, amylase is the enzyme that digests starch into glucose. So when we're setting up this practical, we're going to need 3 different test tubes. One with starch in to be digested, one containing the enzyme amylase, and then some test tubes containing a pH buffer to give us that pH that we'll test. Now bear in mind, in any experiment you have to control everything else that could also affect your dependent variable, which in our case is the activity of the enzyme amylase. So the things that could also affect that enzyme activity other than the pH, which we're changing on purpose, include temperature for certain. So our next step is to take those 3 test tubes I mentioned and put them into a hot water bath to allow them to equilibrate and basically reach the same temperature. We'll use that same temperature for every single pH that we test to control that temperature variable. Other things that could also affect the rate include the volumes of the different solutions we use. So of course we'll control the volume of the starch solution, pH buffer, and amylase solution that we use. Hold on, before we get this reaction going then, how are we going to know the rate of the enzyme activity? To do that, we're going to want to measure how rapidly the starch is broken down by the enzyme amylase. So what you do is, after regular time intervals, you remove a few drops of the solution and test those drops to see if it contains starch or not. And remember your food test, the test for starch is using iodine. So we add it to iodine, and we do that in a spotting tile. If, when doing the iodine test, we see a positive result for starch, that means there's still some starch left in the solution and it hasn't all been broken down by the enzyme amylase just yet. So what we need is a spotting tile with iodine already added to the wells of the spotting tile, ready to add some of our solution to see if starch is still there. As with finding any rate, you also need to know the time it takes for the reaction to take place, because the rate of anything is that thing divided by time. So you're gonna need a stopwatch too. Okay, we're ready to go. We'll mix together all of those solutions, the pH buffer, the starch solution, and the amylase solution, which are now at the same temperature, and get that reaction going. We'll start a stopwatch at the same time. Then every 30 seconds, we're going to remove a drop, add it to our spotting tile, test it for starch, and see if it's still present. Really simply, we're just going to repeat that until all of the starch has been digested and we get a negative result for starch, which is basically when the iodine remains orange. When we finally get that negative result, we'll stop the stopwatch and our experiment is over for that pH. And of course, because the purpose of this was to investigate how the pH affects the rate, we now need to repeat that whole experiment, same exact process, just use a different pH buffer. What we can conclude is that the faster we get that negative result for starch, the faster the starch has been broken down, and therefore the greater the activity of the enzyme amylase. Don't forget to consider how you can